Okay, welcome everyone. This is Christian de Lutz from Art Laboratory Berlin. I'm here with my colleagues Regina Rapp and Tuçe Erel, also Georg Tremmel from BioClub Tokyo, as well as Hideo Iwasaki from BioClub Tokyo and metaPhorest, Waseda University, and Henry Tan from Freak Lab. We have a few other people as well. HyungJun Park from the Media Art Center Route 17. And Elaine and Regina from Fab Lab Bali. Soyoon from the Rice Spring Sisters Club will be joining us later. I wanted to just welcome everyone here to Viral Club Reloaded. It's part of it's going to be an extension of a program we had during the pandemic called the Viral Cloud. And here I can get you a few little bit of a detail on that. We had 14 meetings in 2020 and 2021. It was started just with, that Regina and I, together with Georg, decided that during the early days of the pandemic, we would bring together our two communities. It happened to be that Henry was doing a residency at BioClub Tokyo when the pandemic started. So our first meeting was the 4th of April in 2020. And you can see we had quite a few meetings at the beginning, little bits of a break as the pandemic came and went. A really interesting groups of people who spoke during that time. Some of these are online, like the talk with Peluso or with Hideo Iwasaki and Matthias Rillig, but most of these were kind of unrecorded talks of just between different communities with one or two people volunteering to give a presentation. I think our last community meeting was in spring 2021, but we've been continuing on since then. Henry Tan, for instance, organized last year a BioClub conference in Singapore as part of his residency in Singapore Art Museum. And so I think many of the people were there. I didn't have the good fortune to go, but Regina did, representing Art Laboratory Berlin. And Regina and I were able to visit BioClub Tokyo and metaPhorest in October, November, so which was really a wonderful experience. So the idea of the Viral Cloud was to connect Berlin and Tokyo, but also other locations around Europe, East and Southeast Asia as a forum for exchange, research, and practice. And in that spirit, we thought we would maybe do several meetings this year to bring people together again, and also to connect, extend the connections. I would say maybe, Georg, do you want to add a little something in there, or was one of the co-organizers?
Yeah, just thank you for bringing the viral cloud together again. So it was during the pandemic, of course, everybody was at home, everybody was sitting in front of the computer. So it was very important to make these connections between Europe and Asia. But now the pandemic is gone. So as you can see, the last couple of years that we didn't have any meetings. I mean, we had in-person meetings, which was also great. So I think it's nice to connect again together on the cloud. Yeah.
Okay. Okay, and so maybe what I would do is would start off with a little bit of a talk of what we've been doing in the last time. And I would just suggest for the ease that we would go through the list as we had written it on the website and on the round mail. So maybe Regina and Tuçe will talk a little bit about what Art Laboratory Berlin has been doing in the last year. And there will definitely be some dovetails with later talks, especially June's talk. I don't know, Regina, would you like to start off?
Sure, we can talk a little bit, Tuçe and me, about last year's program, just a few minutes. You see the browser, Matter of Flux. So, I mean, as Chris said, I think it was very, very important after meeting virtually and not having met in person last year. I mean, Tuçe, you can also talk about your residency in South Korea and Taiwan. And as Chris said, I was so utterly grateful. Henry made it possible to bring me over in person to Singapore and Bangkok. So that were moments last year. And then we met Hideo in person in Tokyo and Georg again in Tokyo. So that was really, really, really outstanding. Actually, maybe not even 50 viral cloud virtual meetings could replace a physical encounter. But just to kind of get together all together and just sharing a little bit of a few projects. We did produce a lot, and just for an example, for a little insight. So one of the exhibitions we did, and it was a larger project, it was called Matter of Flux, again with artistic research that we are interested, bio art, hybrid art, that we had invited White Feather Hunter from Canada, actually finalizing her PhD project in it's Symbiotica in Perth, Australia. Lindsay Walsh from the US, based in Germany, and Shuli Chung, based in Paris, with Evan Chardonnay, with projects that basically dealt with biopolitics, female non-binary bodies, health, reproductions, critical ideas of reproductions. And when I scroll down, can you see that, what I see? Yes. So we were very happy to actually have a brand new work for the first time presented. And this is here White Feather Hunter's work, The Witch in the Lab Coat, where she basically used her own menstrual serum, which she found out was ideal for stem cell research, but actually the world isn't there yet. So we had basically a bioreactor with living menstrual cells in the exhibition space, pumped throughout the whole exhibition phase. We had a little bit of a clock in the end, which was very interesting to see as a process, basically. And you can see also here hints to the very patriarchal systems of science as an establishment, which is also coming critique from the artist. But basically here in this beautiful glassware by a glass artist made for a white feather, she had this living bioreactor. Then we had Lindsay Walsh with Self-Care, a project that had to do with a gene mutation that she carries that could have a high probability for breast cancer, and that is a family story as well. And Lindsay would guide us basically through a very intimate story, and also they built a wearable in which there were 2 packages with living cancer cells. And Lindsay would feed them every few days throughout the exhibition time. So that was also very like a bio artwork connected to healthcare, and also very vulnerable points, a very intimate artwork. Maybe Tuçe, do you want to say something about Shuli Chang and Evans Unborn that you also curated? We all curated that show, Chris and Tuçe and me.
Sure. This was a new iteration of this project that was started in 2018-17 when they started the project. But here we showed a new iteration due to certain malfunctions, and therefore Shuli decided to work on the installation a different way. The project's focus is about reproduction, and Unborn is brings the idea of ectogenesis, to have the babies, human babies, to be have a outside the woman's body. So in relation to that, it was focusing on technology, and then how technology is connected with the human body, and then how the reproduction can be rethought from that perspective. So in this work, you see a baby in a artificial womb, and then also a certain device that is planned to scan the progress of the growth of the baby in this womb. And you also see certain keywords that was a kind of a brainstorming idea, which is also a pure data patch, because the project is also developed in relation to ultrasound and then musicians to perform. Yeah. And then in relation to that, we also hosted 3 reading groups that is led by important scholars of their they were focusing on their certain themes. So yeah, you can see the final, sorry for the background sound.
Everything's fine, Tuçe.
Yeah, I'm in a crowded place, so people are clapping time to time. I'm sorry. So we did 3 reading groups about the themes, the 3 themes of the unborn, 0 times 9. They were ectogenesis, ultrasound, and surrogacy. So each session has ended with a live stream, so you can see all those live stream events talking about the reading sessions on our website and on our YouTube channel.
Mm. This is what I'm just showing Tuçe in the permeable bodies already.
Yep. Do you want to continue, Regina, or shall I take over?
Sure. I can just quickly, very quickly, just guide through a series that, as Tuçe said, it was part of metaPhorest. Basically, it is here we had a reading group, as Tuçe said already, and permeable bodies basically was then a series that carried us from spring to winter last year, and it was basically concentrated on women's bodies, non-binary bodies, and going through a metamorphosis under the biopolitics and body politics of the Capitalocene. So we were inviting artists and thinkers to give contributions. You can see here again the reading group, the 3-part reading group. We had Margarita Bevere speaking about the new concept of arts of vulnerabilities, very much connected to symbiosis and body politics. She would talk about politics of uncontainability. And also we had a workshop, as you can see here, with Farah Peluso, Sarah Amanutz, and the biologist India Mansour. And these are a trio with whom we work already for many years in the DIY Hack the Pinke, a group dedicated to artistic scientific endeavors around the river Anke. And they would also go under the term of hydrofeminist ecologies, discovering on a citizen science basis with a broad audience to discover river, but also actually hydroecologies. And we had also podcasts. That was Tuçe's project. And this is beautiful. We're going to continue that, right, Tuçe? This is one of the part of the podcast. Feel free to listen to them all. They're all on our research website. And we talked in these podcasts about menstruation, menopause, and other issues. We had walks, just to make it a little shorter here, because you all want to present and mute yourself soon. But so we had also the topic of menopause in the workshop and in the podcast. And then also we were very happy that we had Mary Magic with us for a virtual lecture and for an online on-site workshop. That was an amazing, a double pleasure. It was Sticking with the Trouble, where Mary Magic worked with a small group on finding objects in urban nature, then bringing them together in a collective procedure, seeing or non-seeing, and then gluing them together with a natural substance, and dismantling the installation later on. So it was a very interesting collective process. And that was actually the the end of our series. And then also we had an amazing workshop on menstruation and decolonizing menstruation, queering menstruation. That gave us all really a big, a lot of input, basically. I will stop sharing, and I already talked way too long. And so thank you so much. It was just a very small overview. Feel free to browse our website or our web pages. We have, as I said, an emphasis on research and a lot of publications coming up this year. So yeah, stay tuned. Thank you for listening.
Right. Thank you. There was also a really good festival connected with Matter of Flux that you can find documentation on our website about a 4-day festival we had in June with local community. I guess I would go now to Georg. Again, I'm just going to go as it is written on the website. Georg, you may could just tell us what you've been up to recently.
Yeah, I have a couple of slides if it's okay.
Perfect.
Let me just check this. So it's just a little bit about BioClub. So those who don't know BioClub, we are really a small but a very nice biolab located in the center of Tokyo in Shibuya, co-located with Fab Cafe Tokyo. And BioClub, the lab itself is very small. It's maybe just 20 square meter. So that's basically the whole of the lab here. But we pride ourselves of having a very large community. And just to make sure the relation to metaPhorest. So actually, I'm originally a metaPhorest member, and I'm still an artist in residence of metaPhorest. And Hideo kindly opened his lab to artists, right? But it's very difficult to bring a lot of people into a university lab. So that's why we have the BioClub as well, where we have an easier place to gather people. And then if they want to make more complex projects, they're welcome at metaPhorest. So Henry was actually an artist in residence at metaPhorest. So just to have one thing which we've been doing now also since 2 years, it's called Mesomics, so meso and genomics. And actually a friend of mine, Atsushi Nida, he's a professor for cancer genomics, and his wife luckily is a very is a kind of a how do you say, a miso? She makes a lot of miso by hand, and the way you make miso is you boil the beans and then you mix it with the bare hands, yeah. And we were asking ourselves, what kind of bacteria, what kind of parts of our human body is actually present in the miso? So maybe the miso I made is maybe different than the miso Hideo made or Nida-san made. So and actually we made miso. And sorry, this is Japanese now. And then actually we did metagenomic sequencing with the miso, and we found so we have the data now. We still need to make sense of it, but we found some differences, and yeah, we actually also preparing a publication for that. So I think this is really very exciting that this is happening in a kind of open biolab. So this is not really an art project, but more like a community learning project. But I think it touches a lot of other things like the non-human body, the others, our relation with bacteria, and so on. And just one thing, I am personally very excited now. Is this, and this is this has been around a couple of years now, but I think right now it arrives in Europe. So this is about the new breeding technologies, NPD, and whether genome engineering with CRISPR is actually GMO or not. Yeah. So in Japan, the clocks are a bit faster in that regard. So we already have a tomato on the market, which is genome edited. They deactivate a gene, and by the deactivation, the tomato produces higher GABA levels. So this is this gamma aminobutyric acid. This is a neurotransmitter, which is also found in chocolate very often. And actually, by eating this tomato, they're supposed to be good for you, But it's very interesting that it's, and actually, I have it in my garden here. I have a little garden, but in my little garden, I have genome edited plants. And what we're doing now at BioClub and also at metaPhorest. So I would like to actually sequence this genome, this genome, and find out which parts is exactly missing, and then synthesize this part, and then reintroduce it to the tomato plants. So by reintroduction, we actually create a GMO, but the resulting GMO will be genetically identical to the wild type. So it's taking this process of new breeding technologies and GMOs a bit ad absurdum. So it's again, it's a learning project. It's still in progress, and I think it was in the news like the other day, at least in Austrian news, was that like the EU level, they decided that there's discussion going on about the new breeding technologies. And it's quite an interesting discussion, because people argue that the NBDs, they are not considered GMO. Therefore, you also don't need to have the checks with it, So it's quite an interesting discussion to have. And another thing, so this is actually Elaine, Henry, so Wave, you're also part of that. So we are now part of a thing called How to Grow. Wait, maybe I need to share my other screen. So this is actually a very exciting online and free course in synthetic biology. It just started now. It's called How to Grow Almost Anything. So David Kong from the MIT Global Community Center, he's organizing this, and he kindly opened this SynBio class, which is running at MIT, to the global audience. And this is really, really exciting, because you have people like Georg, who everybody knows is making a woolly mammoth now, but also other very, very famous scientists like Drew Andy, who coined the term SynBio, Manu Prakash, who's very famous for frugal science, maybe Joe Jacobson. Everybody knows what he did, because he invented the Kindle display. So the paper, the e-paper, the e-ink, he invented 20 years ago, but since 20 years he doesn't do anything in this direction. He just does SynBio now. So we have this amazing list of not only lectures but also labs, which Elaine and me are trying to do now. Yeah, and we might get Open John like robots. So very exciting. Yeah. Okay, so that's all from my part. Yeah.
Hey, thank you. I guess now we'd go to Hideo.
Okay, just okay. Okay, hello, I'm Hideo Iwasaki from Tokyo, and I'm engaged in the biological research and also now making some artworks related with the life concept. And I've already spoken at the BioClub meeting twice, and had a great time with Regina and Chris in Tokyo and Singapore, as I already mentioned. And I'm basically a researcher in chronobiology, so-called, to explore the biological clocks and in bacteria, and so I'm a microbiologist. But before becoming to the scientist, and I had been very interested in the history of science and epistemology, which explore the complicated boundary between science, philosophy, religion, and art, and so on. And this is partly because my parents were scientists, and I wanted to deconstruct them in some ways. But anyway, I'm particularly interested in the struggle between art and science, trying to explore the boundary around the very complicated, how can I say, anthropological concept of life. And so I'm not this time. I'm not talking much about my own project, but I myself have been developed some art projects that could be described as aesthetic deconstruction of biological research. as well as some biomedia art using cyanobacteria and paper cutouts and so on. And I'd like to mention about something about the COVID situation. And under the COVID situation, the Tokyo Olympics was held and exposing a variety of problems. And although I was opposed to hosting the Olympics in Tokyo originally, but I was asked to exhibit some works in an exhibition related with the Olympics. So I decided to show some artworks, which including some intrinsic criticism against the Olympics. But it turns out to be a very unusual exhibition because it's without any audience. So an art show without any audience, any participants, something crazy. But anyway, it was done, and it was a valuable experience, a weird experience. And as for related to some virus station, last year I also wrote a paper, sorry, in Japanese, but on the similarities between the music and viruses. But anyway, anyway, as for short introduction of all the metaPhorest, and as already Regina, Chris, and George already introduced, that in 2007, I set up a platform called metaPhorest as a place to explore both scientific and artistic research to be shared at a biological laboratory in our institution in Waseda University. And as already Georg said, and Georg has been a very long time a member since almost the beginning, and we have supported some creation of biomedia art, and where artists would experiment in the lab, but now it became more like a society for bio-related art. And we have weekly closed meetings to discuss the progress of each project and what interests us. So Henry, so these are some members, and we are now 15 members or so, and Henry and George have been very long supported metaPhorest international activities, and other members include some active artists such as Aki Inomata and Shuko Fukuhara and so on. And we also have recently an anthropologist who is a participant observer of metaPhorest itself. And as for the university politics, we are at the Department of Electrical Engineering and Bioscience, and which is not art department. So, and most of the students I supervise are doing academic experimental biology. However, there are some mutant students who occasionally join the metaPhorest activities and do some artistic researches. And so briefly, I will show them their works. So for example, the Tomoya Ishibashi developed a project that originally reversed the genetics of goldfish and is now more open to be an up-and-coming media artist with a unique blend of AI technology and some craft work. And Tomoki Matsumura, and who is also close to Henry, and used to be a world-class fin swimmer, and is now developing the Daphnia swim, which mimics the swimming style of Daphnia as closely as possible. A little bit crazy project, and Kazutoshi Uemura is interested in relationship between the honeybees and humans, and is now developing a project to outfit bees with the hive masks. And the Eastern Asia also has its very own unique aesthetics of moths, and Kentaro Aki, the newest member, began a project to make clothes out of moss while researching the historic process of moss imagery. That was my brief introduction of metaPhorest. That's all. Thank you.
Thank you very much. And that was really interesting. And also, I want to thank you also for the wonderful invitation that we had to come to speak at metaPhorest in the autumn. Tuçe is going to give an introduction to HyungJun Park, who also had an exhibition with us at Art Laboratory Berlin in September.
Exactly, thank you. So HyungJun and I met in 2019, and then our collaboration extended into an exhibition in 2023, September. So in this collaboration, which was also generously funded by Art Council Korea and then Berlin Senate, we presented Jun's three artworks from his 15-year long career, and we were showing also his ongoing PhD project, Somnoloki, as well as two other artworks in his career, which was an I'm an Artifact and Utopia. So these three artworks was presented between 1st of September to 8th of October at Art Laboratory Berlin. And then I had the pleasure to co-create and then organize this project in collaboration with Juha Lee, who is also with us today as a listener. And so let me also quickly give information about Jun. So HyungJun Park is a Berlin-based artist whose works are based on art and science. His interest includes visual cognition, computer technology, the philosophical self, and the artistics body. His work was exhibited and collected at the Center of Art and Media in Karlsruhe. And then he's also founder and director of Media Art Center Route 17 in Daejeon in South Korea. So after our exhibition, Jun also kindly invited me to do a research residency in South Korea. And then I was hosted by Route 17. So I will hand it over to Jun to talk about this institution that he founded. Thank you for joining, Jun. The floor is yours.
So, my name is HyungJun Park, and I'm Berlin-based artist, and same time I'm a director and founder of Media Art Center Route 17 in Daejeon, South Korea. Media Art Center Route 17 was founded in 2019. I'm sorry. 2019, and then Daejeon is geographically located in the center of Korea. It's a 2-hour drive south from Seoul. In the case of Korea, many cultural events and resources are concentrated only in Seoul. Therefore, Media Art Center Route 17 aim to support local art, local artists, and cultural events. So our center carry out 4 major projects. There are exhibitions, workshop, residency for emerging artists, and then international events. So please let me explain the building structure of our center. It consists of basement and first floor and second and third. Basement is the space for mostly video art, video work. And then first floor and second floor is for the studio and or exhibition place or workshops. And then third place, we have a space where the artists are living during their residence. period. First, I will explain about the exhibitions. I would like to introduce the two kind of exhibition of the solo show. This is the our first solo shows from the artist Jin Soo-jung. He is a video artist and a filmmaker. And this is also second exhibition, our second solo show. Is the artist from Tae-eun Kim. He is also video artist, and then he made immersive video art and then installations art. And next, I will introduce one of the workshop we did. It has the following titles: Exhibition Raum, Living Micro Non-Living Organism. The first workshop we did is about plants. We invite florists as an expert in other field in an interdisciplinary way. Afterward, we went to go to a near mountain with the participants and we collected various seeds. Then we separate the seed in the studio and plant them in soil and water them. And then we move the seed planted by the participants into the incubator. And the process of its growth was exhibited for about a month. The next workshop is about the food and fermentation. I'm excited to introduce this workshop because it's a similar thing like Japanese miso. The presentation of the Gero Termen is a similar miso is Korean word it called doenjang. This is soybean paste, one of Korean's traditional foods. It's a kind of similar to kimchi. It goes through fermentation process. And likewise, we invite a Korean food chef for these workshops. And during the workshop, we talk about the fermentation process and what kind of food material are needed to make this soybeans paste. These two workshops are about organic matter, such as living things and microorganisms. And then next, I will explain the residency program. We invite artists and offer residency programs for 1 or 3 months. During this time, the artist can use accommodations and studio in our center. We usually support young artists. The purpose is to provide them with the environment and then opportunity to focus on their own work. At the end of the residency, if the artists want, we will host a solo exhibitions or open studio event. This is our way of sharing the artist work with the public. Finally, I would like to introduce our international program. We invite artists, curators, and researchers from abroad. I will present today here two cases. First, in 2021, we invite the Berlin-based artist Friedrich Klauke. He stayed here and worked for a month and held a solo exhibitions. It looks like this. And finally, I would like to introduce a curator and researcher, Tuçe Erel. And we invite her in January of this year. She visited an exhibition in our space. And it was the photographer's solo exhibitions, and she also visited his studio. And after seeing the exhibitions and talking to the artist, she wrote a review of his work. Yeah. So here we end the introducing about the Media Arts Center Route 17. And here is the email address. If you have any question, please feel free to contact me. Thank you very much for attention. Thank you.
Thank you so much, Jun, for this very brief but also very detailed presentation of Media Arts Center Route 17. So from here, I want to continue with this residency project that I was involved, thanks to Jun, and shortly hand over to the floor to Soyoon. But I want to also give a little bit information about this research trip, because when it was clear which dates that I was traveling to South Korea, I reached out to people that I know, and one of them was Rice Brewing Sisters Club, which we have great pleasure to work with them in our group exhibition, Hackers, Makers, Thinkers, experiments in social fermentation. And in this exhibition, Rice Brewing Sisters Club did a mini residency in Berlin, and they presented a new work. And so I reached out to them to meet while I was going to be in South Korea. However, the sisters just changed the dynamics, and then they said, hey, we have an ongoing project, and then we are organizing public program. How about you also join our public program in December? So eventually I had the great pleasure to collaborate with them during this research residency that was hosted by Jun and the Media Arts Center Route 17. So therefore, after having this conversation and seeing the exhibitions, current exhibitions, MMCA, and then also Seoul Modern Arts Center, I wanted to include Soyoon in this conversation to talk about their current project and present their Agar Agar kits, and to join Soyoon. I'm sorry, also Soyoon is not going to present this project alone. So let me quickly, briefly introduce them. So Soyoon Roo is a member of Rice Brewing Sisters Club, a collective which has combined artistic research and practice through a framework of social fermentation. Their practice of social fermentation has been hopping between fields of visual arts, performance, creative writing, oral history, decolonial histories, geographical and geological observations, and auntie wisdom. During this conversation, Soyoon will be speaking with Hemin He, an artist and a chef of Andak, with whom Rice Brewing Sisters Club collaborates for the new project Umu Kit. So the floor is yours. Welcome, Soyoon and Hemi.
It's really, do you hear me well? Okay. It's really good to see some of the familiar faces. And thank you, Tuçe, Elaine, and BioClub Tokyo for having us here. Just gonna share screen really quick. It's working?
It's working.
Okay. Okay. So, for those who we haven't met yet, we are called the Rice Brewing Sisters Club, and every year our membership shifts. So this year, we're three, Hemin and Soyoon, myself, and Alethea. And by the end of the year, we gather again and then discuss the extent to contribution, who's going to be the member, how it is operated. So it is kind of a flexible collective / club. And we work around the topic of fermentation, social fermentation, as Tuçe introduced us. This year, just going to introduce our new project that's in the making since last year. And we are coming up with a new entity that is within the Rice Brewing Sisters Club called Rice Brewing Sisters Production. It is both kind of speculative and real entity that engages in these things. First, microalgae, and using microalgae, we are hoping to develop new materials that are both ecologically and financially sustainable. And as a result, create a system that makes the collectives be independent from the grant or exhibition commission dominated system and create more self-sustaining and autonomous financial structure. So we've been kind of making these structures since last year. This is in Korean. We're going to translate this year. That's one of our tasks. This is called the financial ecological model, all based on the broader logic of circulation and redistribution. This is our income accumulation and redistribution model. And this is our collaboration model. So what we're experimenting with Rice Bearing Sisters production is we're not just working as a collective of three, but we are creating a system where there are many other institutions, collectives, and individuals who are a part of Rice Bearing Sisters Club and existing in and out of it. So already we are collaborating with a lot of these spaces, and Tuçe is one of them. Koran, who also visited, is one of them. And this is Umu Kids, our new project that I'm going to introduce soon. And this is Heminhe, who Tuçe just introduced. So I'm just going to give a little space for Rina to introduce herself because she's been collaborating with us for Rice Bearing Sisters production quite intensively now. If you can unmute, she's the chef of Onlop, a really nice vegan sandwich place, and it's still opening right now. So I think she's kind of cooking and speaking at the same time. But if you have a chance, Mina, to introduce yourself. Are you
Hi,
here?
nice to meet you. Like,
Okay.
I think she told everything about me, but yeah. Actually, I'm just like open my shop and like making bioplastic and yeah, like that.
Yes. But her face is going to appear a lot in this presentation. Thanks, Mina. So we are introducing this new product as part of Rice Bearing Sisters production. It is called Umu Kit. And umu is a short form of umukasari, the form of algae that grows in the coast of South Korea, Japan, and Africa, Latin America, and some other parts of the shore geographies. And we use this material to make this. This is what we are right now calling bioplastic, the kind of alternative of plastic materials that are meant to be decomposed easily in the sea or in the room temperature. So after 2 weeks of making in the ideal temperature, it becomes like this, becoming a lot more transparent, starting developing fungi in the process of decomposition. And we are coming up with some super DIY recipes. It's made really easily just with agar that many of you are actually familiar with, being used as the solid medium in laboratory settings with glycerin and water. But it is the temperature and the environment and also the proportion of each ingredient and the duration of boiling it and making it cold, all of that determines the result and the rigidity of and flexibility of the material. So we are still developing the recipe, making this kind of stuff, also making blocks with different food dye, making fabric, and installing at a museum space as kind of a permeable architectural element. And holding kind of a factory production area to make these materials and develop into something usable. And in fact, experimenting with decomposition too. So not just making them and using them, but actively thinking about how it can be melted and decomposed in the natural environment. So we came up with this kit because all of these process starts with the material, and the material is, so these are our collaborators. Mina's here, and her I'm going to introduce very soon. This is the recipe, or this is what contains in the kit. And this is the original material, agar-agar seaweed, that is the red algae. And when we dry it, or when the sea divers dry it, after picking these algae, it becomes white. And these are the sea divers who harvest this algae because as of now, with the current technology, it can't be farmed. So it can only be harvested quite deep in the sea, 30 to 50 meters deep, with the help of the women divers' barehanded labor. And it goes to Japan and the mountainous regions of Japan to be in the miso fermentation site. And it uses the similar facilities to turn them into this form that is called kanten, or hancheon in Korean. And it goes through the multiple freezing and drying process in the rice paddy that are resting in the winter. And Mine and we visited the similar factory in Korea. There's just one factory in Korea right now that produces this kanten using the traditional method. So the result is either this noodle form or this block form that is quite typically used in Japan as food source, but very, very new in South Korea. So a lot of the algae is produced in South Korea, but is quite never consumed. This factory is working to reroute that consumption and production structure. So we've been working with this factory by visiting and hearing their stories. And those materials are in the kit, both from Japan and Korea. And we are pairing up with other collaborators' products. For instance, this is the Wing Wing Home, the Natural Dye Studios product, using chita, also Korean native plant that dyes into this beautiful yellow color. And all of our packaging is also made biodegradably, so everything is gone into the Korean traditional paper packaging using plant dye and coating with beeswax for being waterproof. And this is Mina's contribution, if you want to talk a little bit about it. Here is Mina's contribution in this one too. And we want to kind of use this material not just for bioplastic, for the human consumption, for material consumption, but also edible food, because kanten and hancheon is, at the end of the day, something that we eat. So we also wanted to create recipes and materials that are actively being used to eat. And we're calling agar pudding right now. Yeah, if there's anything to add. This is a syrup. Yeah. The orange ginger syrup.
It's kind of
that Mina
seasonal
made.
syrup for, yeah.
Yes, using seasonal fruits. And this is basically the alternative of glycerin. So we used a similar kind of recipe, but replacing it with the syrup. So it has this tangy and kind of sticky structure that becomes pudding. And all of our kit is accompanied by the recipe that we made together. And right now it's on sale at our museum site at Seoul Museum of Art. And this is kind of the structure that introduces our current Rice Brewing Sisters production business model and operation. And if you follow all the lines through, then you will arrive here. That is the selling station. And it has a little QR code embedded in one of our agar-agar base lumps. And then you can buy this material. Yeah. And our next step is to develop some content around it, because the recipes and also more visually oriented recipes are very, very helpful for learning about how to make this and consume this. So we are making a small community of like-minded experimental artists. This is CSO based in New York, ITP, to develop the shared recipes together and create kind of a recipe video. And we are also pairing this up with different kinds of programs so that our business model is not simply focused on the product, but also the educational creative initiatives around it. Yeah, so this is our QR code. Tuçe and HyungJun here are are 2 customers of the kit already. And if you're interested in looking more into our product, please use the code. And we hope to have more chances to introduce our next videos and activities very soon. Thank you.
Thank you so much. Actually, the parcel has arrived. I need to pick it up today.
Yay!
Thank you very much. Yeah, actually, Riceberg Sisters Club were in Berlin in 2022 as part of the Hackers Makers Thinkers project, which was an exhibition, a symposium, and they did a workshop as well. That was really wonderful. Actually, I would say they had a kind of a residency as well, so they interacted with a lot of gardening communities here, which formed the basis of their exhibition. Was really quite wonderful, and you can find that on some of the links that Tuçe has put into the chat. Okay, I'm really happy to introduce Henry from Freak Lab in Bangkok, who, as I had mentioned earlier, was in a residency at metaPhorest when the pandemic started. So he's been in BioClub since that very first talk or very first meeting in April 2020. Henry's a Thai artist based in Bangkok, and he investigates concepts of life and simulation through the study of technologies like synthetic biology, mixed reality, biosensing, and simulation. And he also has done a series of projects on sleep as well. So I don't know, Henry, are you ready for your presentation?
Yep, hello. I'm Henry Tan. I'm an artist based in Bangkok. I'm a member of metaPhorest Japan and a Freak Lab Thailand. I'll share some of my projects and also a few fun moments that happens throughout these past few years. Okay. So I was running an art space called Tentacles in Bangkok since 2014, and then until 2020s as COVID come, and then I stopped running an art space because I'm broke. And then later in 2000, no, actually I have to. In 2018, I joined a biocamp that organized by Georg and BioClub Tokyo. That was my first entry to like bio art or biomedia art, and then that's how I start to try to learn more about what is bio art is. And then I get a chance to join this global community bio summit since 2018, the second editions, and these events also bring together people who are interested in DIY bio in different aspects, from scientists to artists, from makers to corporates. So we have a wide range of people who are joining these bio summits. That's how I also try to understand how this platform and movement is kind of gearing towards. And then because I was running an art space and residencies in Thailand, so I have an idea that try to kind of gearing the residencies program towards a bio art. Then how can I do that? Is that I joined Freak Lab in 2018. So Freak Lab is a network of artists and scientists around Thailand. So actually the program is emerged from a science camp for elementary school students, and then later after 20 years these the students become professors in universities. And then the founder of Freak Lab, he has an idea to kind of gather back these people to do research, to do crazy projects, to do freak projects outside institutions. That's how they try to initiate different research groups. You can see my screen, right? Okay, sorry.
Yes, we can.
Then, okay, then and then 2022, I got a residencies offered from Singapore Art Museum. Then I tried to expand and overarching it into residencies within a residencies. And then 2020, I host, actually I want to host myself at Pat Pung. He's expert in synthetic biologies in Phitsanulok universities, but I end up inviting 7 friends to join the residencies program, including Georg and a few members of metaPhorest. Then this is the moment, the final moment that Georg sharing tools with Pat Pums. So as an artist, sharing a DIY tools for scientists. So this is quite precious moment. So these residencies also inviting artists and scientists to share their own tools to each other. So we can learn, like with our friends, he also sharing his programming tools. And then our friends also sharing dance tools. And then these are some few moments that where we are feeding elephants after the residencies. We also have performance. We also try to do the agrobacteriums in the setup labs. We also have this swimming, Adapnea swimming workshops for everyone to swim together. And then this is like we prepare before we go into the swimming pool. And we also, our friend Tiffany's also do this invasive cooking with collaborating with the chef. So I'll try to eat all those invasive species. Yeah, we also show some results of different bioluminescence bacteria and yeast. So we have around 50 to 60 participants who are who has no idea about like DIY bios, but now they start to, these movements start to grow in Thailand, which is I'm very happy. And then in 2023, these projects continue into Singapore. We have 5-day open to public workshops, and we are very happy to have Georg, Hideo, Regine, Elaine, and everyone to share their projects, to share their update, and also discuss about different urgency and situations about their own practice and about DIY bio and bio art practice as well. Yeah, few fun moments. So we try to figure out how to use fold scope. Yeah, we keep folding, and then okay, we finally see something. And of course, we endless eating, meeting, experiment, testing our workshops before we launch it to participants. Yeah, this is Georg. Try to prepare this. Yeah, we have different stations for participants to get their hands on, to actually try to do probacterium DNA extractions, bacterial transformations. Okay, then so that's one part of my practice, and I'm organizing workshops and residencies. Then it's also feeding into my practice as well. I also joined this residencies projects in 2021, 22. It's called the sociability of sleep. So these projects bring everyone who are really interested in sleep, and that's how my past few years, my project is gearing towards sleep. Yeah. I've been working with muscle stimulations, VR, lithography, and then this kind of sleeping and resting body start to come into my practice. Then I want to share with you my my projects. This one is the semiconductor dreams. These projects try to talk about like this a hybrid living digital and organic entities that consuming our dreams. Yeah, while we are sleeping, then it's kind of growing itself from our data. These projects kind of try to address these kind of anxieties toward the data surveillance, the tension between the US, China, and Taiwan. But the process of these is becoming a performance that I invite people to sleep through this kind of sound journeys and performative experience. And then another project is the hibernations sleep experiment. So this project also arises from a short research trip with my friends Esther Lu and Huang Tingyun. And we try to understand how human can learn from a non-human body's hibernations, and then how human can perhaps propose a form of a torpor or a form of transformations of our own bodies or our own metabolisms to handle this different climate change, or maybe not only like the climate, but the climate in our societies, in our politics as well. So we create different activities that people can introduce themselves of how they sleep, or learn how they can sleep like an animal, or sleeping together with a pangolin, listen to the pangolin, and feeding your blood to the mosquito. And then another project I did last year is also to create this project called Lanterns. The lantern is the ideas of creating a space where people can idle, rest, and dreams. And then we have these an objects that people can listen to the whispering of the dream stories. Yeah, so we collaborate with students and professors from the University of Toronto, that we try to come up with this space that people can rest and sleep, and then and also that dream can be triggered by some sounds and some textures of our work. So just keep sleeping and sleeping in a place for sleeping. And this is a project that I'm trying to develop further. It's okay. Then it's my current projects that I'll be showing in Taiwan together with Soyoon. It's also about try to create this wearable decay experience for humans. Yeah, that's it for my practice. Thank you for your time. Hope you have some sleep, and then hope Georg have a lot of sleep too.
Thank you very much. Thank you. Yeah, and then our final speaker, and then before we can go, after that, we can go into some questions and discussion, is Elaine Regina, a designer with a major interest in biodesign, synthetic biology, sustainable systems, and regenerative futures. And she's also from Fab Lab Bali. So Elaine Regina.
Hi, first of all, this is my first time here in the Viral Cloud talk session, so I'm really new here. Just wanted to say thank you, Regina, as well as Chris, for inviting me to be here. Yeah, so I actually have a slide to share. So, yeah. Can everybody see my screen?
Yes, we do. Yes, we do.
Okay,
Yep.
cool.
Yes,
So
perfect.
it's the full screen right here. So yeah, as mentioned earlier by Chris, I'm an interdisciplinary designer with many interests, mostly in biodesign, sustainable systems, regenerative features, and radical indigenous and open science movement and regenerative features. How did we get there? All right, let's walk through it. So it all started in 2019. I was basically a design student at that time, and I joined this competition called the Biodesign Challenge. And basically, our team proposes this concept of bioapartment that basically could desalinate seawater for everyday use using this system called the photosynthetic microbial desalination cell that has an algae that could basically withstand salinity because the plan was to genetically engineer it. This is just a concept, by the way, but it's really cool that when we made this was basically inspired by how mangrove trees work. And I just really thought that biodesign and speculative futures somehow they're well matched for each other because at least in a designer perspective, it makes me imagine more positive and sustainable futures. So along the lines, I made a couple of projects that are within that theme. So like this project called Polymono, ideally is like a home kitchen system that could basically break down plastics, polymers back to its building blocks, the monomers that could be repurposed, and then sort of like have an exhibition where essentially the idea is like a creative exploration between food, living, and humans, in particular entomophagy. So people coming in, we will basically ask them, hey, would you like to try some crickets? And then basically imagining the idea of having cricket farms that could possibly power your electricity. So this is just a fun critical concept that we did. And that's where sort of like the line where I started basically like doing a lot of speculative futures. Um, but then going back to the Symbiome project, it actually started because of a real experience. So this is like a massive catastrophic flood that we normally have when in North Jakarta. So I'm from North Jakarta originally. And every time heavy rain occurs, like yeah, I think there was like around 3 or 4 times in my life where I have to get evacuated using these boats because my house got submerged. And essentially, a lot of people are saying that Jakarta is basically going to be one of the first cities in the world that is going to sink in 2050. And that's why we came up with like, can we maybe make an apartment that could desalinate seawater? Why? Because one of the reason is that the man-made natural solution to avoid flooding and rising sea levels is actually mangrove trees. But because obviously because of increasing population in Indonesia, as well as land subsidence and sort of like systemic problem, a lot of these mangrove trees are being cut down. And that's why we're basically suffering the effects of like rising sea level and heavy rain. And that's when I realized, like, okay, it's actually really important to work with nature. And actually, coming to Indonesia, it makes me realize, you know what? We Indonesians have been working with nature a lot. Like, for example, like when we talk about food packaging, we still use this food packaging till today, which is basically this is basically banana leaf. I'm pretty sure in Thailand as well, they use banana leaf for food packaging too. But yeah, like before plastics, banana leaf is always the go-to food packaging. And then if we talk about architecture, like in the indigenous houses in Indonesia are basically built this is all vernacular architecture built with natural material using fibers, stones, basically different indigenous houses has different kind of materials and methods of building it, but they're all natural. And so that's when I realized, okay, maybe if we sort of like want to reach a really positive speculative future, there should be like a really nice intersection of actually accounting ancient indigenous wisdom into the practice. so that we're more mindful about planetary issues. And then I realized, okay, these are, and that's when, like after realizing about that, I started to embed more of those philosophies in my practice. So, for example, this is just basically a concept for a course for an open course called Ancient Future Tech. Basically, the idea like maybe in the future we could have like a digitally fabricated houses with 3D printing that could grow as well as shape trees. And this was like inspired by the living root bridge that you find in India, but there's also one in Indonesia, and which is like it's like a 4-hour ride from my house. So I met the local tribes there, and basically they taught me like how they normally shape their trees, which is so very interesting. And they shape these trees when the trees are still really small and young, but as they grow like 20, 30, 40 years in, the branches will get thicker, and that's how you get the structure to be very, very strong. And then also the idea like some forest tribes are actually, they're so in tune with the nature that they have this sort of like interspecies sort of like communication. And so I was wondering, this is sort of like Garden of Words, is a project that I did for How to Grow Almost Anything that Georg mentioned earlier, an open class from MIT. But basically, I was just imagining sort of like a concept, like a bio art concept, where maybe people could understand what the forest is going through in terms of like, are they under stress? How do forests regulate when they're under stress? So that's a concept that I was thinking of. Also, another thing is this is my work with Georg as well as Nisa from BioClub Tokyo alongside with Tafia. We joined the biodesign sprint and sort of like create this speculative space food concept. And the idea of the space food concept is inspired by the fermentation of koji, the national fungus of Japan, that is used in a lot of like Japanese food. And I mean, like I thought that was really cool. The concept wise, it's really fun to think about positive futures when you mix with like new technology as well as like, indigenous knowledge. But I kind of want to see if it's actually possible to make it applied in real life. And that's when, like I met this amazing person, Manu Prakash, a professor in Stanford who also created Foldscope and championing the frugal science movement. And so basically Foldscope, it is this paper microscope that's probably costing like around $2 to $3. And essentially the idea why it's so cheap, it's because he wants to make the citizen science movement where everybody could get access to microscopes, collect some samples, analyze it, share it with like the whole online community. And that way you build like a citizen science movement to basically tackle planetary issues. He also does some other open source microscopic machines. For example, this is the plankton scope. So you can literally see planktons from your water samples. And so we were able to play around with his machines, which is really fun. And so I met him in Bali, and then we basically like wanted to learn about like the aquatic conditions of the Balinese Ocean. And so we worked directly with the fishermen. We basically told them about the Foldscope as basically how we could tell them like, hey, in a sense you have like lab on the field with the water sample that you collected. You could just check it directly to see if you can find something interesting from the water sample. And then basically, like we found interesting planktons. Like, for example, this is a bioluminescent plankton called the ostracods. They emit bioluminescent lights. This is Acantheria, but we also found microplastics in the touristy areas of Bali. So chances are there's already microplastics in my stomach. And so that's a pretty interesting find. And yeah, and another thing is that I've always been interested in biology, but I don't really have much deep biological knowledge. And so I'm really glad that there's like this open courses like How to Grow Almost Anything, meeting amazing people from the Global Community Bio Summit. And last year, Henry opening these open workshops in Singapore, where I met Regina as well as Henry and a lot of other people in this call right now. So yeah, pretty cool. And so this is also my time in Tokyo. I really wanted to get to learn wet lab protocol. So I went to Japan and told Georg, hey, I'm going to be in Japan for 2 months. Like, let me learn some wet lab stuff. So what is like the red line in all of this is that obviously this open science movement is very important. And that's when I realized like, like for us to reach that positive speculative futures combined with like, indigenous knowledge, I think it's really important to have open science accessible to all to reach the regenerative futures that we want. And so I'm actually like right now in Fab Lab Bali, and I'm pretty glad with these people were actually basically applying those 3 domain of philosophies. And so, for example, like here on the left, we actually saw that Balinese people actually use dead corals as building bricks, and we were just so surprised. I've never seen this, so that was like pretty new to me. And then, yeah, because Bali right now is currently trying to figure out how to reach zero net emission by 2045, inspired by these coral bricks. We're basically trying to like, make artificial coral using biomaterials and also see whether, like if we could grow corals on this, but at the same time use this as material for building constructions or for let's say furnitures and more. Also in Bali, we have this offering called Chanang. Basically, because Bali is a very spiritual place, they still believe in they're very in tune with the nature as well as God. So they do like each household could produce like around 15 of these each day. And usually you would normally make this chanang with coconut leaf as well as this bamboo leaf. But these days, because for efficiency, they use stapler these days. But with bamboo leaf, it's actually biodegradable, but with metals, it's actually not. So for example, if they throw it into the sea, there are chances that the little metal staplers could hurt the turtles and all. So we work with like local high school students, can we sort of like combine this into something modern, and then yeah, creating the shooter with like bamboo lid to make the process easier. And so yeah, right now we currently have a project called the Green Hydrogen Project, the Green Hydrogen Village, because we're currently working towards the Bali Net Zero 2045 emission vision. And so if we talk about like, renewable energy, a lot of people always think, okay, let's make an industrial plant. But industrial plants are quite expensive. And we want to make this sort of like open and then decentralized, make hydrogen cheaper, because right now hydrogen is still very expensive. And so, yeah, we decided to do it in a very distributed approach because we are utilizing the indigenous system called the Banjar system. And we're thinking of like, okay, instead of having a one expensive power plant, why don't we use the indigenous Banjar system to make like innovation hubs with the locals there? And then basically we're going to do community development. And then in terms of the technical research, we're doing this open source and we're trying to make this as frugal as possible. So when we mention about open source, we're actually interested, how do we connect like the global knowledge that could be applied locally? So if everyone in this room who's quite interested in this project, who wants to like, collaborate, we're more than happy to welcome collaborators. And yeah, that's pretty much it. I won't go too long into the Hydrogen Village project, but yeah, that's all from me.
Thank you. That was a wonderful concluding talk. Wow, it really brings a lot of things together that we've had from some of the other talks as well. I see so many synergies here, even beyond the obvious ones that through the network with Henry and Hideo and Georg, but also with Rice Brewing Sisters Club, for instance. I guess now maybe we'll start with the people, the speakers. If you have any questions for one another, just raise your hands and we can work on it from there.
That's great. I have always questions. I have so many questions. Yeah, maybe I would actually fruitful reciprocal question concept is, I would be really curious. Tuçe already said, oh, Henry and June, maybe there's some sleep topic overlap, and maybe you kind of meet later on. Or actually, I see also anti-wisdom and pre-colonial wisdom with the Rice-Bring Sisters Club concept. And I see here Elaine when she says indigenous wisdom. So is there any cross-pollination? Maybe that's too early to ask, but as Chris said, I would be really, really curious amongst the speakers today, those who maybe know one another, or maybe those who have not met yet, if there is some from the practices or the matter that you presented here, because I'm fascinated again about the biomatter that you'll all be entangled with and showed us today, fungi and non-fungi, microbial level, non-microbial level. Is there any question here amongst the speakers? I would be curious.
Or if anybody else has a question, just raise your hand, maybe the easiest way to start. Yeah.
First, I have a concrete question. I know Soyoon, it's so early in the morning for you. Obviously, we cover all time zones now, from day, night times. But if you don't mind, maybe to go a little bit into this, I'm so curious now with the Rice-Bring Sisters production and the new projects. How was your experience lately also to collaborate and to DIWO and maybe also in the post-pandemic phase? However, we are still in the pandemic, no one wants to talk about. But how would you consider the doing together? How has been this going for you?
When we were in Berlin in 2022, the pandemic was quite still going strong, and so the DIWO, as you say, I thought maybe it took place on a microbial level. So what we did in Berlin was we made the compost, the also indigenous compost making method called 섞어띄운 퇴비, mixing and floating compost that is based in rice ball. So when we were making rice balls, then the germs from our hands would be transferred to things that we eat, that we consume while eating, but also goes back to the land by turning into compost. So we did those experiments all the while, but it took place within each project. So every project we had our own collaborators. And what we realized moving on, especially last year, is we experienced kind of 5 years into running a collective, we experienced the same structure can't go on forever. So it's kind of idealistic to imagine a collective to stay the same. I think 5 years were quite enough for us to realize that. So how to also adapt the collective structure into the changing situation for the members, but also how to make the collaboration into not just project-based, but make it into more kind of durational, more sustainable way. So I think those two were the biggest factors for us to rethink the collective operational system. And now the 3 members have been working on the same project, creating the same work, the shared authorship, and we are also this year experimenting with one member working with their own collaborators and finding synergies within those with the internal project groups. So there's a lot of the DIWO methods kind of germinating right now. And I think it's quite natural for a collective as they turn into maybe more quasi-institution or as the structure gets bigger or more grounded, it's just a change that we have to go through. So yeah, and we are also getting really interested in learning about how other collectives cope with these situations. So trying to talk to other collectives about the issues we're going through, etc. yeah, so the issues are not just financial, but also about how to live together in ways that don't exhaust each other. Thanks, Regina.
I have a question to Georg. I mean, I'm curious about the no GABA and GABA tomato situations in Japan or in other countries. Then have you tried it also, and then have you secretly give it to other people to try them? And then also did you get some sleep at all?
Yeah, I'm getting a lot of sleep. It's okay. Okay. Hi. Yeah, but so that's why this is a gray area now about this this fact. What is modified life, and how do we deal with modified life? So and actually, I think last year there was a case in Tokyo where some university students took out Hideo, please correct me if this is wrong, but they took out like modified GFB fish, right? And they for some reason they got up, they sold it on Yahoo or some online store, and this was the first time people were convicted by breaching the Cartagena law of distributing GMO organisms, at least in Japan, right? So I think Hideo, this is correct, right? So
Yeah, that's correct.
So I think if we would bring the GABA tomato to Europe, we might face a similar punishment, like right now. And Korea as well, right? So actually, fun fact: when I was in Daejeon in the Bio Biennale, we tried to show a project called Common Flowers, which deals with genetically modified blue carnations, and we only realized very late that we're not allowed to bring these GMO carnations to Korea. So because the flower company did not bother to apply for the Korean market. So it's quite interesting how this approaches to modified life differs in regions and how this is changing now through new technologies. So yeah.
Nikki has a question. I don't know if this is for Soyoon or for Elaine. The idea of bioplastics from the sea and returning to the sea is quite poetic. However, having worked with marine algal bioplastics, it was found water solubility was a problem. Could the speaker about marine bioplastics please tell us more about the use of wax to improve water resistant properties of the marine algal bioplastics?
Yeah, we so far when we dipped that into the sea, so the blocks that we dipped in the ocean, those are not covered with beeswax, so they're not water resistant. The only material that we use beeswax for is actually the packaging that the Korean traditional paper hanji covered, so that it can contain the food dye or the natural dye. That's the material that we're using. But for bioplastics particularly, we're quite adamant we don't want to add other water resistant material, because one big part of it is to kind of bring back to the sea. And what we've been doing is we make those blocks, but it has glycerin that also is quite water resistant, but is ultimately soluble in water. So when we dip them, it takes about 2 weeks for it to start melting, and around after 4 weeks in warm temperature, spring, ocean, then almost 70% would have dissolved into water. And the rest, what we do is kind of up to the question, but during those times, it develops microalgae around that material. So in addition to being soluble in water, there's also another layer of decomposition happening. So that's kind of how we're dealing with the situation. Another way that is still an experiment is to make them into compost, because we're not sure if this is going to happen. But this is kind of in the works this year if we can also give back to the land. And if we deal with the issue about the block being too salty, then I think we can kind of turn them back to the land, especially the remaining parts that are not water soluble. So we're thinking about that question right now. But yeah, what you say about like not being 100% water soluble is something that we're dealing with. So really, really good question. If you have any idea, please do share.
I think for my end, well, we're not really making bioplastic, although we did actually try using alginate as the binder for our artificial corals. What we do found as well, oh, it's actually water soluble. I mean, we tried using wax as well, but like when we kept it long for like multiple days, yeah, there's still some decomposition happening. And so we're still actually experimenting with that artificial coral material. But what we found, because like we're working with the university-based material researcher, she basically mentioned to us, like, like you can also play around like baking it. Usually sometimes when you bake, let's say because we're using coral, so we're actually using like high concentration of calcium carbonate. They said if you're baking it with several different kinds of binders, you might actually get some interesting results as well, which we really haven't had the time to get our hands on again. And we're trying to like move away from alginate because alginate is quite expensive in Indonesia. So we're trying to figure out how to produce biomaterial, but still at a frugal way, because that's kind of like our motto in Bali. We kind of want to make it accessible to all. So yeah, we're still currently researching this. More than happy to work with anyone who are currently doing this research project as well, working with biomaterials, algae, anything related to sea-based organisms or materials. And yeah, I think here in FabLab Bali, we're trying to build like an open repository to all of our projects. So yeah, that could be something interesting.
We had a workshop in November with Mary Magic, who was working with kuju root, so arrowroot, to make bioplastic. And what she found in the process is it's very sticky. So actually, the workshop was called "Sticking with the Trouble," and she was encouraging people to use it as a sculptural material or as an adhesive to like stick objects together. The other artist we worked with an artist named Farah Peluso, who's also coming from speculative design but doing DIY bio, and has worked with different algae skeletal algae, and the substances on that. And we had her in our Mind the Fungi project, and she used some of the substances from algae skeletons as a kind of making it water resistant, because the problem with the algae is it was totally not hydrophobic. So she made it somewhat hydrophobic, and it was then creating a kind of a fungi textile that wouldn't be damaged by water. Whether you could put it underwater for a long period of time, I'm not sure. I suspect not, but it was yeah, a bit more.
So I
Yes,
think just wanted to add, sorry.
go ahead.
Oh no.
I was gonna ask you
Oh,
a question anyway. Go ahead.
Oh, okay. No, I just wanted to say like we did like an experiment with arrowroot once, and we mixed with the coffee filler. The filler was a coffee waste. And actually surprisingly funny, like me and our friends, we were joining Manu's frugal science class, which he usually holds online. And then we decided to make a cup of coffee made out of coffee waste. And so our filler was the coffee waste, and the binder was actually arrowroot. And we actually found that that was the best combination ever, because not only it's actually waterproof, but it's also heat proof. But I think we actually like managed to like wax it so that like just so that like it's extra strong. But yeah, that's something interesting, funny that we found.
Yeah, and I'm trying to find here the Farah's exactly what she was using. It'll come to me. Let's see. Here's the page from the workshop she did as part of our Mind the Fungi project. So this was a fungal material, but then she coated it. Just see if there's. Yeah. So this was a project. Let me see if I can find the. Yeah. I'll put this on the chat. I think it was a diatomite that she used from diatoms as a substance. It was interesting because the scientists were just trying to work only with in that particular department. It was a project with 2 departments, and that one were just interested in fungal-based materials. So they didn't follow up on that, and they went a different direction. But the other department was intrigued and actually did a project with her with fungi and algae. But that was anyway. The diatomite did have that effect of making it hydrophobic to a certain degree, and yeah. Sorry. And that was part of our Mind the Fungi project between 2018 and 2020, which was together with the Institute for Biotechnology at the Technical University here in Berlin. And Farah Peluso was one of our 2 artists in residence in that project, or in this case, also a designer in residence. Okay, I don't know if anybody has any last questions as we're coming up towards not quite 2 hours.
I have maybe 2 more questions if time allows. I actually wanted to ask Hideo, talking about carrying a concept through many years and really have the stamina to hold on for more 10 years, that the metaPhorest, this is an amazing archive, an amazing polyphonic collection of speakers and contributions, Hideo. Maybe can you tell us how that was for you to carry this through? You say you started in 2007, metaPhorest.
Oh, well, thank you very much. And yes, so 2 years before this, 2007, I got independent and to set up my own lab in a university, and so we need to some new clue to do something new, and so we started several projects, but I was always fascinated by some artistic expression of life or something like that. So I was maybe from 2004 or 5, I started some investigation how life concept is how can I say expressed in art forms. And during the course of that, and I realized the some trend of so-called bio art or biomedia art, and I thought it was potentially fascinating. But sometimes I got bored by some artworks, because some of artworks looked to me at that time that some artists insisted that this is a new form of art, but in reality, some of them were very old-fashioned to scientists. So, but potentially the artistic exploration related with biology is fascinating. So, and I started to do my own biological art practice. But at that time, I wanted to have other artists aside with me to do their own ways to relativize myself. So therefore, we initially employed an artist and who was the painter for the first time, and then decided to open my lab to artists who are interested in biology and life concept and so on. So and actually the first speaker was Georg, and then the platform started. Yeah. And anyway, so it's very interesting, and how can I say, sometimes very frustrating to have the scientific study and artistic practice at the same place, because the culture is sometimes quite different. So, but I really enjoy such a struggle, and I think it's very important. So that's why it's going.
Perfect. Thank you so much.
Thank you.
Thank you so much, Hideo, and good luck to carry this on. This is and I totally
Thank you.
understand.
Yeah,
So
definitely.
also when we say concepts have to be reworked, and then maybe, but some things maybe we can carry through more. Chris, do you allow me one more question?
Thank you.
Because I find, I mean, this is an endless question, but I find I have this impression that in the last 5 to 10 years, academia hopefully could open up or hear or at some certain points to understand pre-colonial indigenous wisdom. This is such a delicate planet, but still, because you explicitly had this in your files, Regine, Elaine, and I was thinking, how could you, if you want to share this with us, because I think it's quite delicate, but so very essential and very important. How would the indigenous wisdom really be carried back into Western normative structures like MIT? So how is it? I know it's a very crazy question, but I think when we want to be there and sharing knowledges, I think it is very important to carry knowledges in all ways. So if you can share with us, or could you make us hopeful, are we there? Can we say that academia would be open to share old knowledges?
So that's really actually interesting. And that's actually the research that I kind of want to dedicate myself for, because, having lived in like multiple countries, I lived in Indonesia for the first 17 years of my life. Then I moved to Australia, lived there for 7 years, but I've also been traveling around too. And then going back to Indonesia kind of make me appreciate like, oh, home more. And then after that, I kind of realized like, wow, there's actually such a huge gap from like, Western technology as well as obviously like the indigenous wisdom that I grew up with. And then it makes me wonder, why is there that gap in the very first place? And I think, as you mentioned, like, well, okay, like a lot of like knowledges in the past, sometimes people are thinking like, okay, this is not scientifically proven and all, but it's really funny. Like, I think the way people say it's not scientifically proven, it's just like, well, I think people just haven't really researched deeply into yet, because I'm pretty sure there's sort of like a science behind it that we haven't really spent our time researching on it. And that's why like here in Fab Lab Bali, we want to like have like this sort of like, in Indonesia we call it titik temu, like the middle ground of like where everybody can meet in the middle. And so that's why like with our hydrogen village project, like we're working with the indigenous system there, but like like not the indigenous system as in like, okay, like you have the system, cool, but we also see how can we use this system to maybe help make like the people that probably people think like, okay, you're not like an academia level, but hey, like you have several other knowledge that could actually be useful for, let's say, like whatever climate crisis that we're facing in. And so that's why like we really like just like open science movement by helping educate like, hey, we have like these cool machines like the laser cutter, 3D printing. What do you think you can do from like based on your knowledge? And actually, like, this is sort of an upcoming project that we're also working at the moment. We're trying to make like a hackathon with Indonesian youth coming from indigenous villages and see if they could utilize this as well. And so, yeah, I think it's probably still a work in the process. But we'll see. Hopefully, hopefully that could work out. And ideally, that's probably my dream, like being able to see multiple indigenous knowledge around the world to be utilized in modern interventions as in an innovation, because I think it's really important.
Thank you. I'm going to ask, because we're coming up on 2 hours, if anybody has any upcoming events or open calls or something that's happening later this year that they would want the community to know about, maybe you can mention that. So just, it can also be somebody in the audience who hasn't spoken yet can also, mention something as well.
I heard that Henry is some preparing something, right? No.
Oh yeah, I'm preparing for this wearable decaying a wooden log. Yeah, try to create these kind of systems that people can experience it, and people can also wear it and sleep it inside the museum. Yeah.
Are there any, I don't know, is there any festival or workshop or labs or camps happening with Freak Lab this year, or?
I mean, I think next weekend we have a small workshop for elementary school students, and Park Boom also organizing a small workshop at the weekend for his student as well. Yeah. So thanks to Elaine that she sent us 70s full scope, and the students will get a chance to try to see things, try to see the world through the full scope. Yeah. I mean, I'm still hosting a few friends is visiting Thailand, so I always like helping organizing and connecting people to whatever they're interested in. Yeah. And like right now, our friends from Austria, Gunther, also visiting. I'm also connecting him to organize some ceramic workshops. Yeah, next week.
Cool. Yeah, anybody else? We'll be continuing this year with with Regina's colloquium and Tuçe's reading club. That will happen, I think, starting in the spring. And maybe also we can arrange some time in the spring another Viral Cloud Reloaded meeting, bringing people together. And maybe
I'm now in the middle
more
of a
focused onto-
deconstruction. Unfortunately, I need to leave the meeting. Thank you so much for joining, but please continue maybe another 5 minutes, but I need to leave the room. I'm evacuated here. So
Okay, I think
I'm
we're
so
gonna
sorry.
close up.
Thank you,
No
everyone
problem.
that I have met in South Korea and Taiwan. It was so great to see you all virtually. Thank you for coming.
Great. Then I'd say, yeah, if anybody wants to add something in now, otherwise we'll close. And I want to thank everyone who spoke for taking part, and I'd like to also thank everyone who came and joined us as well. You can send us emails as well if something comes after, an idea question comes later, or we can pass it on, and probably in about 2 to 3 weeks we should have a recording on the website.
Yeah, thank you, everyone. Meet soon, hopefully.
Thank you, Bart.